And I can finally record. Thank you everyone for coming. We're really happy today to have Sarah Kemp. Sarah has dual affiliations with University of Tasmania, and we have quite a few people from University of Tasmania with prior on current affiliations with University of Tasmania here in the audience today, as well as with University of Amsterdam. And yes, Sarah is a postdoc now, and she's looking at response inhibition and motor control across the lifespan. Today, she's going to tell us about some new work where she's looking at proactive and reactive inhibitory processes, where she quantifies these via changes in corticospinal excitability and behavior and how they vary as a function of aging. And she's also going to talk about Bayesian model averaging and the utility of Bayesian approaches in brainstem research. I guess before we start, I'd like to acknowledge the traditional owners of the lands on which we meet today. Here in Perth, we are on the lands of the Whadjuk Noongar people. I'd like to pay my respects to the elders past, present, and emerging. And I also like to pay my respects to Aboriginal and Torres Strait Islander peoples where you are at, where you're on today. So without any further ado, I'll hand the mic over to Sarah. Yeah, thank you so much, Sarah.
Cool. Thanks, everyone. Thanks, Leanne, for the nice introduction. Thanks for having me. So yeah, as Leanne said, I'm a postdoc at University of Tas and the University of Amsterdam. So I'm also from Tasmania originally, but I've spent the last nearly 10 years living sort of back and forth between Tasmania and the Netherlands during like my master's and my PhD. So I'm very lucky to have been able to do that. Um, so yeah, so just wanted to provide some context about where I actually am. So I'm coming to you from the Netherlands. I actually live in Utrecht, which is city, basically the city next to Amsterdam, in the center of the Netherlands. So where I come from in Tasmania on the left looks kind of like this. This is near where I grew up, and this is the city that I live in now. So I'm very lucky to sort of be able to live between two different worlds, very different places, but both beautiful. So today I wanted to talk to you about one of the last papers that I did for my PhD. I finished my PhD about 6 months ago or so, sometime last year. And during my PhD, I was focusing mostly on response inhibition. So this is the cancellation of a planned or ongoing movement. I know in different contexts, different literatures, it will be used for other things like the interruption of thoughts or the interruption of speaking. But the context today is about stopping motor action, so interrupting movements that are ongoing. So this is the kind of thing where if you see a traffic light and you have to stop suddenly, if a traffic light suddenly turns red, you might stop. Or if you're biking and you see a dog, suddenly you might stop. Or if you're driving, you see a child, etc. So this is a very important part of motor control. Obviously, it's very important to be able to see things suddenly appearing and respond to them effectively and in the right way. But this gets much worse in older age and also in many clinical conditions. So this is a very broad research area that's studied in many different contexts, many different clinical conditions. So oftentimes this is studied using the stop signal task. So I'm sure many people here have are familiar with the stop signal task, but the basic idea is that in every trial an arrow will appear pointing left or right, and the participant responds with the left or right button press. And then in some trials, a stop signal will appear shortly after the go. And the idea then is that you try and not give a response. So you try and stop your response. And it's normally possible to stop the response about 50% of the time. That's kind of the idea with the stop signal task. And what we generally understand with response inhibition is that it seems to occur kind of via two stages. So when a stimulus appears that's kind of telling you to stop or modify your movement, there seems to be this kind of generic suppression across all limbs, so all like effector muscles, where they will suddenly suppress and then the movement will be cancelled. So you get this kind of generic sort of knee-jerk response where everything suppression is reduced and then the movement is cancelled. I am sort of assuming that everyone is familiar with the idea of like muscle suppression and measured using TMS, so I haven't explained that too much, but a lot of this work kind of comes from TMS and from brain stimulation. So there's a couple of key papers that show that when you get this sort of stimulus appearing, then you get this kind of generic suppression across different limbs. So in these sort of papers, they're looking at leg muscles while people are having to stop their hands, and they see this kind of suppression when the stop signal appears. This is another paper that shows this. So they got people to respond with the stop signal task using their feet, and then they found that in the hand muscles they would find this suppression. So it's very global. That's the idea. But a lot of work in the stop signal task is done in very simple contexts. So there's a lot of animal work, for example, which, or obviously the tasks have to be very simple and straightforward. Generally, the requirements for this task is that you have to stop all movements. So you have to either go or stop in any individual trial or in the task. And it tends to be limited to just left or right hand button presses, so quite simple movements. Um, but some people have tried to add more complexity to try and make these movements sort of slightly less simple. So this is one version of something that you can do. This is called a response selective subsignal task. So here, instead of just going with one button, with one hand going left or right, here people go with two hands in every trial. And when the stop signal appears, they need to stop one hand and keep going with the other. So you get this kind of split into a unimanual movement when the stop signal appears. So trying to get a little bit more of a complex movement. It's also thought that this is a little bit more better related to real world movements. Like it's not often the case that when you have to stop a movement, you would stop like completely. You're more often kind of adjusting actually rather than stopping entirely. So this is thought to be kind of a bit more of a real world task. And you tend to find in these tasks, you get this sort of stopping delay in the ongoing hand. So when people have to just stop one hand and keep going with the other, the hand that they keep going with tends to be much slower to respond. So you get this ultimate response delay. And it's thought, well, who knows why this actually is, but it's thought maybe this is happening because you first do get this sort of global suppression, and then that hand has to recover in order to keep going. So that's kind of the idea. And there's been quite some work looking at how you might be able to get rid of this stopping delay, what you can kind of do. This is some early work from Frederick Bruckner and Adam Aaron that were looking at what would happen if you would cue the participants to give them information about what they might have to do. So in this task, they told the participants whether they would have to stop with the right hand or stop with the left. And here they found that people were able to minimize this delay. So it's still there, but they are able to minimize it. And in this sort of case, you don't get that same global suppression across all effectors. So when people know a little bit more about what's coming up, they seem to be able to modulate these effectors a little bit more specifically. Is that all clear so far? Is that all good? Okay, cool. Thanks. And then sort of the song of my PhD was like, what are about older adults? So a lot throughout my PhD, what I was really doing was taking a lot of this kind of work and looking at how it was applying to older adult populations. And we know that they tend to be much slower in stop signal tasks. They tend to be a little bit less adaptive. They have longer stopping delays, all the sort of stuff that you would expect. But why was not so much understood. So this was a big question throughout my PhD. And what for this particular paper, we wanted to try and understand how they are actually preparing for movements compared to older adults, to younger adults when they have informative cues. So how do they utilize these kind of cues, these sort of maybe stop right cues compared to younger adults? Okay, so let's have a look at what we actually did. So we took two groups of adults. We had a group of younger people and a group of older people, and we got them to do one of these response selective stop signal tasks. So they're responding with two hands, and they had to respond. They had to stop one of two hands in the stop trials, and we had informative and unformative conditions, which I'll show you in a minute. And we used TMS to try and look at corticospinal excitation throughout. So this is the task that we had. So in all the go trials are bimanual go, so people are pressing with both hands in order to respond. And when there's a stop trial, they had to either stop the left hand or the right hand or both. So this is what you see in that third column. So the red arrow represents what they should have to stop. So there's sort of three stop conditions, if that makes sense. And we gave them either no cues, so where we didn't tell them anything about the trial that was upcoming, or we gave them informative cues where we told them what kind of stop would be required. And these informative cues were completely reliable in terms of what kind of stop would be required, but it would only be that case 25% of the time. So say it said maybe stop left, that meant that if the trial said maybe stop left, 75% of the time they would have to go, and 25% of the time they would have to stop left. But it would never be the case that if it said stop left, they would then have to stop right, if that makes sense. So the cues were reliable. And then we were using TMS at four different time points, and these were all during the movement preparation. So we showed it, we did TMS at the point of cue onset, and then at three time points just before the stop signal onset. So all in this sort of movement preparation period. So we wanted to see how older and younger people would differ in terms of preparations to stop, what effect the cues would have, and how older and younger adults, older versus younger adults would be able to prepare these effectors kind of specifically. All right, is that kind of all clear so far? Any questions? Okay, cool. All right. So I apologize for the people for whom it's like 4:30 p.m., but I'm going to talk a little bit about statistics now. So I'll try to keep it lively. So we use linear mixed models to analyze these. I assume everyone's kind of familiar with linear mixed models, but the idea of these is that you are modeling every single person on the individual level rather than on the group level, and we use Bayesian linear mixed models specifically. The linear mixed models are really, really helpful for analyzing basically all data, but this kind of data particularly, they're really, really good for unbalanced designs. If you've got missing data, unequal group sizes, you're going to get an improvement on your fixed effect estimates. And with Bayesian LMMs, why they're so useful is that you can quantify evidence for and against the null. So I think for a brain stimulation context, they're super, super helpful. And if I, yeah, I sort of assume everyone's familiar with them, but if not, these are some really good papers that you can have a look at if it's a newer sort of idea to you. Okay, but with mixed models, one of the downsides to them is that they're very, very complex. So because you have this kind of flexibility in your model structure, it means that the amount of ways that you can specify your model is there's a huge number of variations in terms of how you can actually do this. So if you think about things in terms of like your random effect structure, you could only have random intercepts, so like a simple model. You could have random slopes for some of your predictors. You could have random slopes for all of your predictors. You could do interactions for these. And all of these kinds of choices adds to the model complexity very rapidly. And it also means you get just many, many different types of model structure. This is also true for the fixed effects. Like, do you include all your predictors? What about your covariates? If it's a regression, do you only do a linear term? Do you have a polynomial term? Etc. So there's just like a huge model space really um, for the possible structures of your models. There's many, many opinions and many, many discussions about how you should go about selecting your model. So what do you do to get the sort of optimal model structure? There are people who think you should go for the simplest one. There are people who think you should go for the most complex one. A lot of people will use things like AIC or BIC to compare the model fits and choose the one with the best model fit. But what you're essentially having to do in this instance is you have to make pairwise comparisons between all your models. So does this model fit better than that model? Does that model fit better than that model? Etc. And the sort of difficulty with comparing your model fits is that these just tell you which one is better, but they don't tell you about model uncertainty. So if you've got basically two terrible models, one could still be better than the other, but they're still both terrible, really. So that's kind of the difficulty. You don't really get an idea about how good the model fit actually is. And then just because of this like variation in the model structure, it can just get really confusing. So like, what do you do if you've got under one random slope structure, you get your fixed effect, like your effective age or whatever that's preferred. But then if you check that model structure, that random intercept out or that random slope out, then it's not preferred. It can get really, really complex and to sort of be trying to make a theoretically meaningful decision and try and make sure that you're assessing your model in a way that actually makes sense can get very convoluted and you get really tied up in knots really thinking about what it is that you're actually looking at. So for this paper, we use something called Bayesian model averaging to try and get around this a little bit. So what Bayesian model averaging does is that it takes all of the models that have an effect, so say it's your fixed effective age, and it compares all of those models, the posterior probabilities of all those models to all your models without that effect. So you're comparing all the models with an effect to all the models without an effect, and you get a Bayes factor for the evidence of including that structure that effect in your model against across all the models. So, for example, you could have your age thing, so you could compare all of the models with an effective age against all of the models without an effective age, and then you get a Bayes factor for evidence of inclusion of that factor. And that's kind of regardless of whether you have random intercepts, random slopes, a fixed effect of this other factor, a fixed effect of this factor. So it lets you get a really clean look at whether or not that fixed effect or that random effect should be in there in general. So it helps kind of clean it up. It sounds very confusing, but you'll see in a minute, and you end up getting really, really clean results because you're just looking at things sort of overall, and you're able to do away with a lot of that complexity when you're interpreting it. We're using Bayes factors here, so you end up with a ratio. And if you get a ratio of, if you get a Bayes factor of higher than one, that means you've got evidence for including that model. Lower than one, it means evidence against having that factor. So I think I'm messing up the words model and factor. It's very early for me, so I apologize if I'm messing my words up a bit. I've only had one coffee. If you're not familiar with Bayes factors, I kind of assume everyone is, but that's the sort of thing that you're generally looking at. So generally, the value of the Bayes factor tells you like how you sort of weight that evidence. So the higher the Bayes factor, the stronger the evidence, basically. This is absolutely not our idea. This most of this work is based on some work of Daniel Heck's in this top paper in computational brain and behavior. So we were really looking at this paper quite closely to do this, and then all of our code and how we actually did this is all on OSF. So you can have a look if you're interested. Okay, so let's go through some of the results. There's a lot of Bayes factors in this paper, and I've just picked out, well, there's, and what we found is that the Bayes factors tended to be extremely, extremely high, or basically extremely, extremely low. So we have like very extreme evidence in all cases. So I've just picked out the things for which we have very extreme evidence. So I'm not going to talk about like what values the Bayes factors are, but if I'm mentioning it, just know that the Bayes factor is like 1,000. I'm just not going to list them all, but it's very, very high Bayes factors. Okay, so let's talk about the stopping delay first. So this is that effect of when you have to stop one hand, how long the response time of the ongoing hand is. And we found that of course, as everyone else finds, you get this increased reaction time in that ongoing hand. So that's that slower hand, little turtle hand on the left that I've got. We found, as you would expect, older adults had a much longer stopping delay than younger adults, so they were very delayed. And in the informative condition where people were told a little bit about what stop they might have to do, everyone was a bit shorter, but not completely shorter. So they were able to shorten that response delay a little bit, but not entirely. And we found very strong evidence against an interaction here. So although the older adults were slower, everyone actually benefited in a similar way from the cue. So it's not that older adults had a different level of adaptation using the cue. That was actually similar between older and younger adults. Is that clear so far? Okay, cool. So I'll move to some of the TMS results now. So this is where we're looking at MEPs. And for reasons of model complexity, we didn't look at all the different types of TMS conditions in one model. So we split them up into sort of theoretically meaningful comparisons. So I'll go through those now. So first we'll look at this guy, which is comparing the conditions where people might have to stop the left hand and might have to stop the right hand. So what we want to look at here is how well people can actually modulate the specific effectors. Now, with this setup, we're only measuring MEPs in the left hand. So we only had the coil over the right hemisphere, and we were just measuring the left hand. So everything is with respect to the left hand. So I'll try to be really clear about that. So if people saw maybe stop left, that meant that the measured hand might have to stop, right? But if they saw maybe stop right, that would mean that the measured hand would never have to stop. Okay, does that make sense? So if it's an MSR condition, it means they will absolutely have to move with that hand because you would never stop the left hand in an MSR condition. So here we're comparing basically what happens when you get the signal that this might have to stop versus what would happen when the hand wouldn't have to stop. And here we saw a really big Q difference. We found that there was a really increased suppression when it was signaled that the effector might have to stop, and it really, I guess, facilitated or decreased suppression when it would not have to stop. But a really strong null effect of age here. So the way that older and younger adults actually did this was similar. So the effect of the Q was really strong, but we definitely had no age effect. So no difference between older and younger adults here. Now we wanted to look at the difference between global and selective stopping. So what would people do when they might have to stop both hands compared to what they would have to do when they would only have to stop one hand. So we're comparing the MSB and MSL conditions. Here we saw that overall older adults would show more suppression, so that's sort of regardless of the cue. That you would kind of expect. This is a sort of more proactive, sort of cautious response from older adults. And we found that in the selective condition where they might only have to stop that one particular hand, everyone had increased suppression, so this more sort of selective suppression, but again, no interaction effect, so no age difference in terms of that global and selective effect. And then finally, we wanted to compare the condition where we had no cue at all and the condition where people might have to stop both, so a sort of a preactive and reactive switch. And we found that again, older adults showed more suppression overall, that's kind of effect that you would find. And then when there was no cue compared to when there was a signal that you might have to stop both hands, there was no cue effect, so no difference between the suppression. So we drew from that that people were only using the cue information when it was really helpful, because if you might have to stop both hands, it's not like in terms of selectively modulating, there's not really much point to that. So the cue information was really only used when it was actually giving you information about how it might be useful to modulate your hands specifically. Okay, so participants were able to use the cube to effectively suppress their CSE. Their modulation was really effective. They were really good at it. It was really nice. We really found throughout that older and younger adults were utilizing these cues in a similar way, which surprised us. So these effects were really, really big, and there was really, really strong evidence for the null effects here, which sort of caused us to argue in the paper that this behavioral decline that you see in older adults is probably not caused by some difference in the CSE modulation. So this sort of preparatory mechanism, whatever this is, that they're doing before they actually start to go. In our case, in our task, it seems to be preserved in older adults. We really didn't see any sort of decline for the older adults. This is the first application of BMA, Bayesian model averaging, to brain stimulation data. And I think this is a really, in terms of using linear mixed models, it's a really nice way to assess your data. And I really think that Bayesian approaches can be really helpful in cases like this and really with brain stimulation in general to help draw conclusions about null effects. So thank you for your attention. Thanks to my co-authors. I did most of this with Nick Stevenson, who's in my lab at the University of Amsterdam. And so thanks very much to those, and the paper is out. Thanks.
Oh, apologize. I was saying thank you so much, and I'm just trying to open the floor for questions before I realized that I hadn't unmuted. Do we have questions? John has a question.
Yes, I do. So, hello, Sarah. Thanks for your talk. Um, I've seen this sort of information before in relation to response inhibition and aging and how it's slower. Um, is there anything that we can do about it? Is there a rehabilitation protocol or a training strategy that you can use to modulate response inhibition?
Yeah, thanks, John. There's definitely been a couple of papers that have showed that you can improve this kind of response inhibition performance tremendously. It does require, as you might expect, a huge amount of training, like days and days of training. There's one particular paper I'm thinking about that came out about 10 years ago, I think, where they were basically able to optimize the performance completely. Would say though, my sort of as being like a neuroscientist by training, this is a reflection of degradation in like the white matter and degradation in the connection. So even if you are able to improve their performance in the task, I wouldn't say that that's going to generalize in any particular way. So I think there are definitely there are individual differences in terms of how much people will decline in older age. So there tends to be some differences in people in like their 50s and older 60s and 70s, it seems to be from connections between the subcortex and the frontal lobes. So the strength of that connectivity seems to basically inform how well people can do this. So I guess the thing when we finally figure out the best way to like improve your myelination over time and to stop that kind of declination, that will be the thing that helps this. I think there's obviously all the I know there are studies where people have gotten people to bike. There's some like Parkinson's studies where they get really good intervention effects using getting people to cycle and that kind of thing. So probably all of the standard like exercise-based interventions are probably going to be very useful. But eventually, I think we're going to have to try and figure out how to keep connectivity or stop connectivity from degrading in the subcortex, which I think is going to be a difficult question, I guess, over time.
Okay, and so I'm also wondering whether there's any differences between upper and lower limbs in relation to response inhibition with age in particular. And I'm thinking about the sort of left field idea of um, older people who drive and they're selecting brake and accelerator, and I don't know if that's a response inhibition problem, but obviously they select the accelerator instead of the brake, and then they don't take their foot off the accelerator until it's too late, and then they crash. So are the effects the same in upper and lower limbs, and is that situation a response inhibition problem or a response selection problem?
This is a perfect question. Are you I feel like you're applying to an audience member? This is what I'm doing in my postdoc actually. So I'm now in working at Neura and Beck St George's lab where we're trying to literally put these tasks in the lower limb, so in stepping, getting people to walk around and do them. And we've at the moment, basically, I could tell you what we have at the moment is that the data are really, really noisy, and it's been really, really hard to try and figure out what on earth is going on. And at the moment, we're trying to see if these kind of classic findings actually replicate when you take people, when you start making people walk around and step rather than sitting with their hands. Because I completely agree with you. The idea of sitting at a computer and how you respond, and then in sort of the real world with driving and that kind of thing, it's like we always use driving to be like the thing that we're actually studying, but it's actually realistically quite removed. So I don't know what TBC, I guess. We're at the moment, it just seems very noisy, and it seems like some people, like yeah, some people are going to be cleaner than others, as you would expect. I think there's also another issue with motor control, which is that we tend to select, you tend to get this sort of natural dropout. So people who are worse at the task in general are probably not going to end up in your study. Maybe they will drop out themselves or they won't sign up if they have motor control issues. So I also tend to wonder how much in like our health sort of healthy studies of older adults we have this sort of selection bias. So maybe our top sort of 25% of the population or 50% of the population. So I suspect that they might actually be worse, unfortunately, than we think they are. Both because we've got these city computer tasks, and then in the when we get them to walk around, they seem to be quite not as good. So yeah, I suspect that they probably are, yeah, much worse. I think at swapping this kind of movement, because also their decision making is a little bit slower, which is also going to be a problem. So to actually decide what they're going to do, which is also a problem. So yeah, good news.
Okay,
Yeah,
thank
we'll wait
you.
to see. Yeah, we'll wait to see what the results of that are like maybe in a year or so. Cassandra has a question. Okay. Cassie, yeah.
Sorry, yes, my bad. Thanks so much, Sarah. I really enjoyed that talk. Um, I was wondering, because I've seen some sort of reaction time studies, not so much with stopping, but with starting. They might use startle, and startle can sometimes evoke out that response uh, in advance of what they could have otherwise. And we've used startle, and generally there's been a consensus that the brainstem's involved with those sorts of things. And I just wondered if you thought that how the brainstem might be involved in this case, or if you really think that this is primarily a cortical mechanism or a cortical degradation that might be happening, or yeah, what your thoughts were there.
Thanks for the question. I think it comes from the brain, from the subthalamic nucleus, probably, so from the subcortex, the deep brain. A lot. There's a lot of early work in rodents that showed this, where they have like invasive electrode. There's some work by like Robert Schmidt, for example, in like 2013, 2017, that shows this pretty nicely. And so what they find there is that you get this kind of pretty consistent response from the subcortex when you get this kind of infrequent stimuli. They're using it in terms of stop signals, but it probably does work for anything startly, anything infrequent, I would suggest. So I think probably. I'm, I'm trying to pick a side. Do I think it comes from the cortex or the subcortex? Yeah, I think it probably comes from the subcortex, I guess. I'm not sure. I'm not sure. I'm sort of in two minds about this. You get kind of there's differing evidence about the temporal signatures. There's some yeah, there's always some DBS that shows stuff like the other way around. So yeah, I'm still in terms of where they actually originate, I'm not sure. I kind of assume it has to be the subcortex because it wouldn't really make sense that such a fundamental response would originate from the cortex. That means it would have evolved much later. So I assume it's in the deep in the brain. Um, I also have a little bit of beef with that whole literature, so I don't know. I don't know how much to get into it, but um, I think there tends to be a bit of a sampling issue in that literature where they are looking at particular brain regions and they identify the way that those particular brain regions are associated with this, which isn't the same as looking at all brain regions and seeing which ones are the most associated. So there's a whole literature around like the STN and that kind of thing, but we're also measuring the STN. So um, yeah, I'm not sure, but yeah, I think it comes from the subcortex. Is my answer, I guess.
Yeah, no, interesting. Thank you.
Any other questions? I might ask yeah, I guess I'm kind of surprised that this is the first time this Bayesian model averaging approach has been applied to brain stimulation research, and it kind of makes me excited to try to do that for, say, behavioral motor learning data that we have. I was just curious to hear about what you thought about what are the best use cases for this within brainstem.
Oh, that's a nice question. Thanks again. I think anything where you have complexity in your task, so anything where you have a lot of variables. So say you've got, I don't know, a flanker task with different congruency effects, or if you've got like a group condition, maybe you've got like three groups or something like that. So anything where you've got more than like a two by two design or something like that, really. If you're doing a linear mixed model and you start to sort of set up your structure in different ways, you'll see it really blows out. So I think that can be where it's really, really helpful is anything where, yeah, there's just a lot of choices that you could make in your analysis structure. I think that's where it's going to be really helpful. If you've got a simpler design where it's or a really theoretically strong idea, that's really obvious what you want to be looking at. But often I think experiments tend to have sort of multiple sort of little bits and pieces to them. So if you've got kind of multiple components, then I think it can be really useful. But if you've got a really strong idea about what, say you've got, I don't know, a single manipulation and you've got like a clinical group and a healthy control, then your research question might be quite clear. But if you've got sort of like a group of younger adults and then you've got like sort of maybe three or four manipulations, then it can be really helpful, I think.
So say in a more complex design, say in a design where people are trying to, attempt, say, a precision neuromodulation approach, for example, um would that be feasible? Is it also like computationally quite expensive? So like the analyses that Wilbur has attempted with us, not Bayesian model averaging, but yes, some of that does take a while. What has been your experience with this? Is it computationally expensive? Is it actually not an issue?
This wasn't too bad. So relatively, I will say I do a lot of cognitive modeling as well. So like I say, it's not too bad, but also I'm happy to run something for days. So I think, let me think about how long this took us. Probably 2 days in total to run all the models, including yeah, maybe a day to run all of if you didn't make any mistakes and thought it was all fine from the beginning. Um, I'm trying to remember if we ran this on the server or not. No, we didn't. It was
Or
just on a local,
just on local
on a
PC?
Mac.
Yeah. All right.
Yeah. So it is on it was on Nick's Mac, which is one of the sort of one of the more beefed up like new chips, but I still think it's going to be very feasible. We just ran this in R, so you could run this, like all our code is on OSF, so you can just run it. And I can also just send you our data if you want to just sort of completely replicate it and see how it works. So yeah, I think it's very, very feasible. Some of this particular data set is very, very noisy because we have like older adults doing brain stimulation. So if you have sort of like a younger group, I think it's going to be much faster. So we really had a lot of noise, which is quite kind of why we moved to this in the first place, I think.
Do you have any further questions from the audience? I'm just going to kind of scroll through it. Sometimes I can't see everyone.
I can ask a quick one, maybe.
Oops.
Thanks, Sarah. I just wanted to piggyback off that last thing you sort of said. And what do you think is that noise when it comes to collecting brainstem data in older individuals?
Well, um,
There's
that's
probably a lot, but like from
what
your
is noise?
experience working with other
Um,
individuals and collecting EMG
yeah.
and MEPs and stuff like that, like is there anything we can do to better minimize some of the noise?
Thank you for that great question. It's a really great question. Um, yeah, so most of my experience is I've done a lot of work with older adults. I've only done two TMS studies, so I don't have as I have a lot of experience. I have a lot of data sets with older people. Um, TMS I'm less experienced with. So there'll be other people in here who know more about me in terms of things you can do with the TMS data. Specifically in terms of things like gain and that kind of stuff. And I know Mark Hinder, who was my supervisor, he has a lot of tricks for trying to get people to be still to try and reduce that noise in the actual muscles, that
Mm.
relaxation. Just forcibly telling people to relax while he's breathing down their necks, that kind of thing. Um, yeah, older people are difficult because they tend to be quite tense when they're very worried about they really have that lab coat sort of effect, so they tend to be really worried about performing well. What we find, I am always really emphatic to them, and I find this does help in terms of that it's fine to make mistakes. One of the problems as well in the stop signal task and in response inhibition is you want them to mess up because you need examples of cases where they've failed, and that can be really hard to eke out of people. So being really emphatic can be really helpful, I think, to try and get them to not to relax a little bit. Um, but yeah, so you have noise sources from the TMS machine itself. You have noise sources from their behavior. You also have older adults are very, very variable. And if, especially you're talking people in their late 50s to early 70s, you get this kind of split in terms of some people aging really well and they are no different from someone in their mid 30s. And other people, if you've studied older adults, you'll have seen, you see someone who's like 65 and you're like, oh, see their date of birth and it sort of gives you pause. So you get this sort of big variance in terms of how people are actually aging, and those people who are aging, sort of who are showing that they're aging much like kind of worse, for want of a better word, those are going to be the people who are really noisy because they don't perform the task really well. They're going to physically be weaker. So in terms of TMS, they're going to have more movements, but their responses are also going to be really, really slow and sometimes really fast, and that creates a lot of behavioral noise. So you have sort of
Mm.
yeah. I really think we should keep those people in there though and try and work our analysis around them. I really don't like cutting those kind of people out. So like in my others papers, I always try and keep those people in and do a lot of tricks with like using log transforms and that kind of thing on the RTs to try and keep them in because I really don't like this kind of cutting out bad performance
Mm.
in older people.
Yeah, because I was just sort of
Yeah,
thinking
what's your
along the lines of because I know even when you haven't got people doing complex inhibition tasks and just asking them to stay nice and relaxed and rested so that you can get true resting MEPs, that can be really challenging. So,
yeah.
like, did you consider maybe the pre-stimulus EMG, and is there a way to maybe account for that? Does that explain some of the variability? Do people as they
It's
get older, sort of, you mentioned it's
good
a bit harder for them
good point.
to to stay relaxed? Does that sort of manipulate the map a bit as well?
Yeah, nice one. So it's all relative. I didn't say this actually, but it's all relative to that first Q. So we normalized it according to their average Q1 excitation, I guess you would say. So that was kind of the purpose of that Q. So you could also use baseline. I know people do that, of course. So that's what we chose to do. Is that we wanted to try and just get we just chose that because it's in the task. I know sometimes if you use like a baseline, you get reviewers saying like, oh, you could get like just generic task excitability. So that's what we chose to do. So we normalized it according to excitation at Q onset.
Okay.
Um, yeah, so that's how we did it. I don't have any strong opinions about the best way to do that as long as you pick something. So I think baseline
Makes sense, yeah.
EMG is really is good as well. So yeah, that's what we did. Can I ask a question to everyone else?
Yeah, go ahead.
Is that okay? I'm curious to know if people like use Bayes, like what you think about Bayes. Like, would you, for people, especially for people that do brainstem, which I think is most people here, like, do you use it? Does something stop you? Like, what do you think about this like philosophical idea that it's sort of the way to get around the kind of I would sort of argue it's the way to get around the file drawer problem, that it's like a really nice way to write a paper that says like we got a null result and we've strong evidence for it. Like I'm curious what
I
if there's
love
anyone who
that.
disagrees with me or
I love it. And yeah, in the lab where I used to work in Dux's lab and Hannah Filmer's lab, yeah, they published a few registered reports that show strong null effects, and that's very useful for the community, right? So those were using the aggregated approaches, not the disaggregated
yeah.
approaches, but still helpful. I mean, those were published a few years back, but yeah, still super helpful. And yeah, I'm glad to see people applying more sophisticated approaches like what you've done. So we're gonna have a crack, I think, on using these approaches, not just with brainstem, but other types of behavioral approaches where you have lots of variables or lots of variable data. I will note though that in some fields, yeah, people do still like to default to your very standard aggregated t-test, and we'll still see that in say conference presentations and or when you're reviewing papers and so on. And yeah, my default response usually in the reviewing of processes, yeah, please quantify this p-value
Mm.
with the various factors. You did still please do it.
Yeah, I think it's really like even just like a Bayesian t-test I think is a really nice way to look at these data. Maybe I'm just being young and optimistic, but I feel like it's kind of a way to almost get around a little bit the file draw problem, where if you've got sort of a strong null result, you can publish it and write like a strong case, which I think would be really cool. I was interested because I was at a really interesting seminar at ACNS last year in Melbourne about brain stimulation, and I don't know if anyone was there, but they were talking about the this issue of it being so hard to get reliable results out of brainstem, and they were talking about kind of the reliability issue. And there were just a lot of different ideas about what we should actually do to to improve kind of reliability. And I sort of go for a statistical perspective, but other people were talking about like shared data and open science and bigger data sets and all that kind of thing. So sort of in my mind about what we should actually do about this kind of problem. I think it's interesting.
Mm. What about other people? I think quite a few of us already use Bayesian statistics. Maybe not Bayesian model, not Bayesian per se.
Nice.
Yeah. So, do you have any other questions? I guess yeah, maybe I'll throw like a kind of related question out there just because we do have a bit of time. Um, I'm humbugged. I found aging research to be honestly a little bit too hard because of variability between individuals. There's so much variability in the way individuals age. Um, I was wondering maybe John and Eric and other people in the audience also can chip in. Um, what do you think is like the big direction in terms of aging and aging optimization research? Like, is it stay in Australia and maybe elsewhere in Europe? Because yeah, Sarah can tell us a little bit more about that. Um, are they are funding agencies actually working hard to try to work through this issue where we have massive population aging? Other approaches that we have championed, say in Australia, are they appropriate? Are they fit for purpose? Do we need to be looking at more innovative, more souped up brainstem approaches or other types of approaches? Do we need multimodal approaches? Sorry, yeah, very long rambly question. If anyone
No, it's a
could,
good question.
yeah. Yeah, what are your thoughts? What would be your wish list for governments to fund, given that we're all, marching towards this rapidly aging society?
So yeah, I'm really curious to hear what other people think who've got more. I've worked with a couple of like these kind of bigger grants where you're trying to have like this big consortium that's trying to like solve some aspect of aging. And I find it's okay in terms of like getting the money, but then in terms of the questions that I can actually solve, I find they're I don't know, honestly, they seem pretty diluted to me. Like it's just really hard to make. There's so much pressure to answer so many aspects at once and to have so many different groups sort of involved with it that I like in terms of the actual result that you get. Yeah, I'm really curious to hear what other people think really who've worked with like I know there's some people who've worked with sort of bigger um yeah, these kind of bigger collaborations and like longer term grants. It's just like I don't know.
Yeah.
I'm curious what other people think. Um,
Yeah, John, do you have any thoughts?
Um, Look, I really think that we probably need to work towards more longitudinal aging studies, but clearly the funding model doesn't support that because, you want to track people as they age through a period of time, and your funding runs out in 3 years, and therefore you can't really track them very well in that period of time. And so I think longitudinal studies are necessary, but no one's really willing to fund them to that extent. And I also think the issue was brought up earlier about the variability in the way people respond to aging. And obviously, some people respond well and others don't. And we don't really quite understand a lot about how that works. And we also have a bit of a sampling bias because the people who don't age well don't want to participate in these studies, so it makes it very hard to do. So, yeah, trying to understand the differences between a successful ager and an unsuccessful ager is probably important, and how they change over time. So, again, does a successful ager at 60 remain successful at 70? I don't know. Um, I assume so, but trying to figure out why that is probably important.
yeah, I guess first, are these turning points just appearing in different time points for different people? So is your
Yeah,
60-year-old going to be the same at 80 or something? Yeah,
Yeah.
yeah, I
yeah,
completely agree.
Yeah.
yeah. Are there like multi, because I'm assuming people know more than I am. Are there these types of big, ambitious, multimodal approaches, like say in the US, where they quantify things like microbiome, and they quantify they have, I assume, large data sets where they have structural integrity data for in these types of longitudinal designs. Do these exist? I feel actually they must do somewhere. Maybe like through veterans, like the US veterans types of research structures. Oh, yeah. Maybe
Yeah,
not.
Yeah, there's some
yeah,
here.
I don't really
Oh,
know. I think they do exist. The challenge is getting funding for it because you want to try, well, government agencies want to focus on disease. They don't necessarily want to focus on successful aging. So it's very hard to sell that sort of grant to get it funded. Whereas, you've got someone who's had a stroke, it's very easy to sell that to a government to get funding rather than saying, hey, we've got these old people who are healthy, but we want to try and figure out why. It doesn't have the same kind of impact. I think that's my view. So it's
mm,
very hard to sell.
yeah, I agree. I think also there's a couple of, well, there's at least one study that I know of here that's kind of doing that. But you have to be there's another issue with like when you're trying to do structural scans and that kind of thing is that you have to be really clear from the beginning about what your research question actually is because you have to make sure the data that you're getting is actually able to answer the question that you are looking for because things change so much in like MRI in 5, 10 years that some of yeah, these scans they just tend to be very diluted and very hard to actually answer something like these questions about like the STN, for example, that we were talking about before. You couldn't answer them with these kind of scans that exist as far as I know. So yeah, you have to somehow be someone that can write a really strong argument to a government like this is worth it, and this is the research question that we have, and have a really strong um, hypothesis from the beginning, and have really good ideas about what you're actually going to measure. Because I don't know, my experience, they just take so many measurements that it over like 5 minute blocks that it actually getting sort of like good quality data. It's diminishing returns. I would
Mm,
say.
yeah. Yeah.
I think, to be honest, I think short term maybe a good approach is to start looking at things more on the individual level, to take individual people and try and look at how predictors that we can get from them, how this informs their personal task performance. So things like their structural connectivity, how they can personally adapt. So yeah, I'm getting increasingly interested in trying to look at things really on the individual level and see how individual things predict sort of trial-wise performance or individual performance. That's my sort of short-term answer, but longer term, I don't know.
Yeah, and getting those getting better neurophysiological measurements and also getting using better statistical methods that account for your or help you predict the predictors of outcomes. Yeah, yeah, definitely. Do we have any other questions from the audience? Yeah. But yeah, so I kind of yeah, I wanted to wrap up by thanking Sarah again for sharing this really cool new work because I think yeah, it has utility, at least for me, learning about this has utility, so I really appreciate it. And yeah, again, if you have any people you'd like us to reach out to speak, let us know, and then we can try to organize that hopefully at a time that works for most of us. Thank you so much again, Sarah. Really appreciate it.
Thanks, everyone.
Yeah,
Thanks for your
thanks,
questions.
Thank you.
Thank
Thanks
you, I
very
really
much.
appreciate it.
Thank
Thank you.
you. So I'm just going to end the meeting now. Thank you. We'll hopefully see you in the next webinar with Xianwei Che, who used to work with Paul Fitzgerald. Yep. Thank you. All right. Thanks, everyone. Bye now.
Bye.